Hi everyone, welcome to my channel Pure Biology, and today I come with another video obviously related to the MHC complex. So one second. I'm checking the video is properly seen from the camera or not. Okay, before going to this topic, I want to say thank you to all of you who have already subscribed my channel and try to share this channel with your friends if this channel is helpful to you. So today's topic is the major histocompatibility complex related topic. That means how this MHC complex working, or how they work in the processing or the presentation of the antigens. So there are two pathways. Generally MHC molecule follows two pathways. One is the cytosolic pathway and another is the endocytic pathway. And here I will try to describe only the cytosolic pathway. So you just carefully learn. So here, what is the actually cytosolic pathway? Cytosolic pathway means the antigens. These are synthesized in the cell, within the cell. So here the endogenous processing, or the processing and presentation of endogenous antigen actually seen here. Okay. Endogenous antigen is seen here, and here the antigens were produced. They are produced inside the cell. Okay, inside the cell, and they are derived from pathogens. Okay, pathogens means bacteria or virus, etc. that have infected the cell. So what happened here? First the cell is infected by the pathogens, and then toxic antigen that is released within the cell. Okay, clearly understand that. First the cell is infected by the pathogens, and after that the toxic antigen that is produced inside the cell. Okay, and this processing occurs. So we know within the cell there is two parts. One is the cytoplasm and another is the nucleus, and there are many organelles in the cell that remain within the cytoplasm. And here the main organelle, that is the endoplasmic reticulum, has the main function. So first what happened? The major mechanism for generating the peptide fragments in the cytoplasm is via giant protein called proteasome. So what happening here? First, peptide that is already produced within the cell, it is fragmented by one enzyme. This enzyme is known as the proteasome. Okay, first this is fragmented. This is fragmented in the proteasome, and this cleaves the proteins into peptides about is amino acids. So what is saying here? That first the peptides that is produced within the cell, these are fragmented by the proteasome, and this cleaves proteins into peptides about 15 amino acids. Okay, so each fragment must be with 15 amino acids, and the fragmentation is possible by what? That is the proteasome. Clear? And another remaining fragments that are the not like 15 amino acids. So these are actually removed by the aminopeptidases. These are the enzymes present in the cytoplasm. Clear? So what I am saying here, that which are not containing 15 amino acids, these are totally removed from the cell by the aminopeptidases. Okay, these are the enzymes also, cytosolic enzymes. Next, what is saying now? The peptide antigens are transported via an energy dependent reaction into endoplasmic reticulum. So now what is the actual amino acids happen or what is happening with the amino acids? These antigens or the 15 amino acid containing the fragments, these are transported energy dependent reaction. That means here energy is obviously needed. Without energy, ATP or ubiquity, without energy this transportation is not possible. So it enters in the ear through the pore formed by the proteins called the transporters associated with antigen processing, okay, or TAP. You can say TAP, TAP. So what is the name of the pore? Sorry, through the pore, the fragment that enters into the ER, and this pore generally formed by one protein. This protein is known as transporters associated with antigen processing or TAP protein. Clear? And once peptides entered in the ER or endoplasmic reticulum, they bound to MHC1. Okay, they bound to MHC1, and that is just remain near to TAP. Okay, that is just remain near to TAP. Okay, next I think there is the pore. Sorry, 1 second. I think the pore is known as, here explanation is wrong, that means I think the pore is known as TAP, okay, not protein, okay, pore is known as TAP, clear. That's why the MHC1 that is situated just near the pore, okay, near the pore or TAP site, clear. And by a group of chaperone proteins, generally, the name of the chaperone, some chaperone proteins, these are the calnexin, calreticulin, and tapsin, and ERPS7. Okay, and there is actual role of chaperone protein is seen. The MHC1 peptide complex is released from the chaperone and moves to the cell surface, where it integrates into the membrane and can be recognized by the TC cell. So, by a group of chaperone protein, 1 second. So, here actually the role of the chaperone protein is what? Actually, they help in the binding of MHC1 with the peptides. Okay, actual main function of the chaperone protein, it helps to bind the MHC1 molecule with the peptide. Okay, so this is the possible, the formation or the complex formation is possible only by the help of chaperone protein. Clear? And what happens? If the binding formation is completed, that means when the MHC1 and peptide complex is already formed, then these from the chaperone protein that is actually helping this formation, these are released. Actually, this chaperone protein, these are released from this complex. Okay, and what happened to this complex? This complex try to move to the cell surface, where that it integrates into the membrane and can be recognized by TC cells. Okay, it goes where it goes to the cell membrane, and then it is recognized by the TC cells. Okay, and this processed antigen known as the agritope. So this is the flow chart how this is possible. Endogenous antigen, then with the help of ATP, cytoplasmic proteasome complex is formed, and then peptides are formed, and then peptides that by the help of TAP it enters into the ear, and then after entering into ear the peptide MHC1 complex is formed, and then what happened? It moves through the cell membrane. Okay, cell membrane or cell membrane surface. Clear? Cell membrane surface. recognized by the T cell. Okay, so this is the actual process of the cytosolic pathway, and I will try to describe about the endocytic pathway in my next video. Okay, so I think you all properly understood about this cytosolic pathway, or the reactions, or not reactions, activity, you can say activity of MHC1 with this endogenous peptides. How is it possible? This is explained here. Clear? And in the next video, I will try to describe about what happened to the exogenous peptides and the MHC2 complex. Clear? So, thank you.